Today we're talking about turtles, so tracking turtles and testing water, new tools for an old conservation program. Over these last few years, since I've been with the RCD, I've been trying to look at new, different kind of techniques to explore how we do monitoring, how we do surveying. And so two ways in which we've gone about this is using some new technology on turtles and then also eDNA, which that'll be the second part of this presentation. So starting with turtles, we have been doing now for the past 4 years, geez, we've been using new technology, GPS units, to track turtles. The reason why is that turtles, I'm not sure how many of you guys know this, but they were a candidate species currently to be listed by the US federal government. That's taken a bit of a pause with the current administration, but we're hoping in the next couple of years, we're going to have these listed species. So in Northern California, we have two different turtle species, or in California in general. We have the northwestern and southwestern pond turtle. They're currently recognized as two distinct species. That may change, interesting enough, with some genetic work we've been doing. But the idea is we want to start learning better what they're doing and where they're going. Because one of the issues with turtles is that turtles are considered they're an aquatic species, but they spend a lot of their life on land. And where they spend their lives on land, we don't really know as much about as you would think. Um, for instance, they actually lay their eggs on land, and then those eggs sit underground, actually hatch underground and sit underground from between, call it June-ish, until the following spring. And so these turtles are actually kind of living a lot of their lives underground on upland areas. And not only that, turtles also move across the upland. So males especially will move fairly long distances because they actually will travel from pond to pond or river to pond or across watersheds or whatever they are doing to try to find females or even just to spread, to move from their natal pond to some other pond to kind of keep that nice genetic flow going. So our project goals were initially to track turtles to better understand their movements. We also wanted to, and that's using GPS, so I'll go through that in a second. We also wanted to look at female nesting locations, so we paired up with the University of Sonoma State to do that. And then we wanted to use all this information we gathered to inform on conservation strategies. So here's a nice little picture of baby turtles. Our project sites, these keep growing. As I said earlier, Ben Weiss, he just joined in with the East Contra Costa Habitat Conservancy. We're now doing work with them. And so our project sites now are across a lot of Alameda County, City, Livermore, and Pleasanton. We're working in the urban creeks there. We have Contra Costa County rangelands. We just started working in Morgan Territory and Clayton Ranch. And then we also now work with SAPUC on their creeks and rangeland. So our sites keep growing. We also, part of a larger group, we also are working in San Francisco. We have groups working up north. We have groups working down south. So this project really is growing quite quick. So what are we doing? Two different things I mentioned. So one, we are using radio telemetry, and this is kind of a more, call it a older technique. And so VHF is what we kind of refer to it as. This allows you to, so you attach a unit to a turtle. This allows you to track a turtle throughout the landscape using like very old school. If you think about like those people like hunters and all that stuff, using like the old school antennas, trying to find the animal. That's what we're doing. So we're actually capturing females as they're coming out to nest, quickly putting a VHF logger on it. Usually they return to the water, and the next day they try it again, and we're actually able to locate where they're nesting. This is really great technology. However, you have to be present. So the people that were doing this are master students who have way more time and are way cheaper to use than professional biologists. So they're the ones out there daily, really trying to track these animals. And I'll share some of the data we collected later. And this we're using primarily, or actually just exclusively for breeding. So this is the way in which we can find active nests. The other one, which is a brand new technology that we've started using a couple years ago now, these are the GPS units. You can see it on the turtle in that picture. Sorry, it's a little small. These are really cool. They're way less than 20 grams. They're solar powered. They connect with either Bluetooth, or we actually use cellular towers to connect with them. So these things actually connect and give you almost like a live update. You can almost track a turtle in real time. These are great because they work as, again, as far as being less expensive, I can just log on to my computer whenever I want to, pull up where the turtle is, download the data, whatever it is. It doesn't require me to be there. So it's kind of like a plug and play. Have it put it on the turtle and let it collect the data for you. And it's great because so far the units can last almost up to 3 years. We find the units kind of around a year and a half seem to get some waterlogged, but we've have some that have been on for 3 years, which is kind of incredible. Only issue is they are hard to get back. So once they're on the turtles, you have to try to catch the turtle again, which that's the only thing we're trying to finding that's a little hard is that a couple turtles are kind of elusive. So we're still collecting data from them, but we do want to get the loggers back and off them. The loggers do not cause any damage. They are stuck on a single scute. So the turtles themselves, we've seen them breed, we've seen them mate, everything. So they're not like it's causing a damage to them, but it's still good to get them off their shells at some point. So where are we currently? These numbers are so, sorry, I did not update these numbers. These are so not the right numbers because we are well over 40 loggers now. We have about a split equal, I'm just going to fast forward to that because it's not what it looks like, but you can see video instead. Um, so we are actually up to over 40 turtles. We have them in urban creeks. We have them in livestock ponds. We have some in rural creeks as well. Um, we have a fairly equal split of males and females. It'd probably be about 20-20 or maybe 25-15 with a few more males because I've been very focused on male movements more than female. And so it's really going well. And the loggers continuously just pump out data. I mean, it's been every logger has lasted at least a year and a half. So we're getting this really consistent data across what we've been finding. So important, all this data is still preliminary. We're still about to actually, we're in the review process now. So I just wanted to put that out there. Okay, so for this presentation, to make it kind of fun, I want to focus on just male movements because we're going to go into female nesting movements next. And I'm going to focus on a couple turtles. Um, so it's three males and Urban Creek, three males in 2023. This is the ponds that did kind of crazy movements, and then an Urban Creek male. And the reason why I want to focus on these is there's a lot of kind of misconceptions or assumed things that turtles do. And what I thought was so interesting about what our research has revealed so far is just how little we know about their upland movements. Based on a very small subset of turtles, we're able to see some things that you should not be able to see from such a small subset of turtles, really kind of revealing just how little we know. So, male one, we call him the lazy boy. This was what I'm kind of going to show you is what the experts say and then what we've seen. So, male one, lazy boy, what the experts say is males typically stay in situ during breeding. And this was a full year of movement. And so, as you can see, this guy sits and does absolutely nothing throughout the entire year. He goes just to the border of the ponds. That big part of the middle is a turtle raft. So he sits out there. That's all he does. And this was our first year. And so we were actually only put loggers, GPS loggers, on 5 males in total. So again, I'm showing you 3 of those males. And so this was our kind of classic turtle. This is what you expect a turtle to do. It sits there. They kind of stay in the pond. Don't really do much, barely even leave the pond. Maybe one of these points could be it sitting out or overwintering, but for the most part, this turtle just hung out in the pond the entire year, only traveling the distance of the pond, so that 82 meters. So really kind of that lazy, lazy classic turtle. Next one I want to show you is we're calling him the pond hopper. So what do experts say? The U.S. Fish and Wildlife studies in 2020 say that turtles prefer in-water movement, but they can go fairly long distances. And not only that, but they have a max distance of about 500 meters from a pond. So from this, you can see we have our turtle pond here. This is a livestock pond. That's where the little lazy boy sits. And then we have another pond here called Ridgefield Pond. This is on Patterson Ranch, the Zone 7 property. And the turtle fairly easily goes between the 2 ponds. And again, this is something that we expect. We expect them to be able to move through drainages and get to ponds. And the interesting thing is, the max distance from a pond back in 2013, okay, that's 500 meters. Okay, we're stretching it a little bit. All of a sudden we're seeing, because we have these loggers on these turtles, we're able to see this actual distance change. So instead of being 500-meter distance, guess what? This turtle can go over a kilometer or roughly a kilometer. And so you start seeing these kind of, we're kind of pushing that idea now. This is the pond hopper, he's hopping about, but it's really showing that although we expect this, turtles can quite move fairly far by themselves. They don't necessarily just sit like the lazy boy. But it still matches roughly what the literature says. Next, we have the city boy. Now, this is a turtle we have in city of Livermore. And this one was really interesting. So again, pond hopper went a little further than we expected. The city boy even stretches that. This was a journey this turtle took in about a week, I believe, was the total, but actually made the journey from the endpoint down to here and back. It made that in about, that was, I think, a couple days, 2-3 days. And so, again, we're looking at what the experts say is that a 10-year study, the farthest traveling turtle was 5 kilometers. And somehow we put loggers on not that many turtles. So in the pond system, we only had 5 loggers. On this creek system, we only had 10. And yet we're sealing a turtle within the first year, really hitting that record. So wow, it's going to 4.5 kilometers. So this turtle actually traveled from this is Livermore. So it's actually able to travel through the urban creek all the way through here. Actually went up to a culvert that was blocked with some cattails, then traveled all the way back and actually surpassed that distance. And now he's actually gone even further to the other end. And this again, the study was the first year we were able to see this movement. It's just kind of incredible. Again, we're kind of testing that limit of what we think turtles have the possibility of doing. And then finally, we have our extreme. So what the experts say, again, that 500 meters max from a pond, and they prefer water movement. Well, this is a little different. So this is a turtle. This is our first year. He started moving around March. We're calling him the Wanderer because this is a turtle pond where that lazy boy started. And he decided at some point to take a massive journey. And this is a drainage that at the time was actually primarily dry. So he went through a drainage, stopped here. Instead of continuing with the drainage, he actually decided to climb a hill. And that hill, which I hope I have some scale of it, but I do not, is ridiculously steep. I hiked it. It's over 100 meters high with a steep grade. Traveled across that, spent about 3 days resting at the top, and then started his journey down. And so at the time, when we actually lost our GPS reception, he had traveled by just in a straight line about 1.7 kilometers, so probably about 2 kilometers if you calculate in the curve. And we thought we were done. GPS reception went off. We don't have reception, unfortunately, down in Del Val. So Del Val Lake is just over here, just to the right. And so we thought we actually lost our turtle. However, thanks to a nice camper at Lake Del Val, they found the turtle in the creek, caught it, and actually notified one of the rangers at East Regional Park District. And so we were able to find that that turtle actually took an insane journey. He climbed up a hill, down another hill, went down into Lake Del Val, swam across Del Val, an entire different reservoir in a different watershed, and then went up Del Val River and was actually saw Arroyo Del Val and actually was caught right here, this last little point. So that little image, if you take it for what it is, I mean, it's harder. You could say how the crow flies. If you did that, it should be probably more impressive. But his little journey was probably a minimum of about 7 kilometers. And so again, we're pushing this whole thing. We keep thinking these simple things we know. It's in the literature saying that farthest they're going to travel is about 5 kilometers. And in the first year, we collected data from these turtles using GPS units. We're able to see that this turtle was actually able to travel about minimum of 6.5 kilometers. And it's very likely that he kept going because unfortunately, those people, they released the turtle, so I was not able to see it again, which is a bummer, but we got photos and documentation, but it would have been great to actually pull the data off the logger before they released it. So that turtle very well could have continued swimming up into N3 property. So the whole purpose of this is really just to show you, you know, what we think we know is not what's actually the reality. And that when you're actually able to utilize this new technology, you're able to see that these turtles are doing some very cool new extremes. Potentially this idea of these abnormalities, this idea of pond hopping, traveling over 500 meters, using only waterways to travel. Maybe those are not the exceptions, and actually maybe this movement, this turtle going 6.5 kilometers, maybe that's more of the norm. Because if you only have 5 loggers, again, we had 5 loggers in this one pond, and two of the turtles did something we did not expect. And then with the turtle in Livermore, we only had 10 loggers, and this one turtle did this massive journey. So I think we know a lot less than we actually think we do. And I think this new technology is really trying to begin to reveal what's happening. So jumping over, again, with these GPS loggers, something else they begin to reveal is we wanted to see how well they identify nesting behaviors. Now, none of this, again, this is very preliminary. This is stuff we have not proven because we can't find the nests, because if you know anything about turtle nests, they're almost impossible to find. But we wanted to see if we actually use these to identify nesting. And in this image, you can kind of see, so this is a turtle who, during the breeding season, during June, came out of the pond, went about 33 meters from the creek, hung out during the exact times in which a turtle should, so between about 4 o'clock to 7 p.m., went up into the area where they should be nesting, did something. Obviously, you can see all the points. And then the next morning, stayed overnight, and the next morning returned. And so what we're finding is that these loggers not only can they detect movements of where they're actually going and show us their live locations, but they also can potentially show us nesting behaviors. And so what we're finding is, again, this is something that unless you had students sitting in the field observing nests on every single hour, watching the turtles, really being focused on them, we would have never have seen this. And so these loggers are beginning to show us things that we did not think possible. If they're actually showing us locations where the turtles are nesting, which is really important for conservation practices as for turtles being able to protect that nesting habitat, which you can see is right near a road. It's quite important. So being able to identify roughly where they're going, what conditions, what habitat they like for nesting can help actually protect their nests and maybe even their overwintering. Okay, so some initial conclusions from this study. Turtle movement's really complicated. So what we think we know, what the literature says, may not be the limits of what their abilities are. And in fact, I think it's more to testament to there's not enough papers and not enough information on turtles. And so we're basing a lot of our conservation practices and controls on literature that is not focused on the general population, but single isolated instances. And so the more we study, the more we know, the better conservation practices we can put in place to help protect these turtles. In addition, they have GPS now because not only can they alert us to where the movement is, it's also allowing us potentially to see behaviors. So what happens, we're able to see now potentially nesting opportunities. Can we actually use these to determine where they're overwintering? So turtles will brumate underground during the winter. And can this actually help us identify what habitat they prefer brumating in, and therefore understand where we should protect them when they're also brumating? And then finally, what about females? Again, we saw that nesting stuff, but I'm going to go into this next part of this topic, next part of this presentation, to really look at females specifically. Any questions here? Because I'm going into a very different topic now with VHF. Okay. So nest site selection. I'm going to do a little more reading here because I have this actually presentation is from a student of mine who just finished up her master's, Maddie Stein. And so this is her full study at the Sonoma State University looking at nest site selection. So first to start, what is nest site selection? Nest site selection is a non-random placement of nests / eggs by a female turtle to maximize their chances of nesting, successfully producing offspring. So the aim for a turtle is to optimal nest site selection and to minimize the mortality of females and maximize the reproductive success. So pretty much they want to try to find the best place to nest, not to just reproduce successfully, but also not to endanger themselves. This is the extremely critical behavior in turtles, especially since there's no parental care after eggs are laid. So once turtles lay eggs, it's done. That's all they're doing. It's also the only chance a female has to pass her genes on is to successfully select a nest. So not only is it important for her to pick the certain spot, but also in addition to it being, potentially a spot that does not get predated, it's also critical because turtles use thermal and moisture conditions to shape the embryo development, sex ratios, and habitat traits. So therefore, if it's a hot site, you get female. If it's a cold site, you get males. So really picking a specific spot in shade with moisture, all the different conditions, the right temperature, and the depth of it will actually influence the sex of the turtle. It also influences the risk of depredation and inundation. So the chances of hatchlings successfully reaching water and even the survival of nesting females will all part of this whole thing. So again, this is a really important thing. As you see from this little, the nice image she created here, the figure, it just shows kind of the importance of everything that goes into nest site selection. Okay, so now jumping into the field season. So in 2024, Maddie and some volunteers tracked and exposed 20 nests and recorded nest characteristics. The green are the successful nests. The red are the unsuccessful nests, the failed nests, which means that the turtle actually went out of the water, dug a hole, but never actually ended up laying nests. This spot again is Patterson Ranch. This is where the lazy boy was, the kind of the pond I keep showing. This is a unique site because from the get-go, you just think it looks like a normal cattle pond, and it is just a normal cattle pond, but the soil type is extremely hard. And what we found was super interesting is that turtles were really struggling to dig through it. And so we actually had quite a number of failed nests, and also even successful ones, they wouldn't necessarily plug the nest itself. So once a turtle crawls out, she digs her burrow, lays her eggs, then what she'll do is she'll urinate and then cover it with a plug. And we found the soil was so tough in this area that she wasn't actually necessarily successfully able to do that on a lot of the attempts. Still, the eggs were quite successful, the nests were, but it's an interesting characteristic. Um, so the first female here uh, had a nest, was also the last female to nest. So on June 6th, that was the first nest. And then on June 26th, she laid a second nest. And so that's called double clutching. It's one of those things we thought was not necessarily super common. And we're finding out from this study that it actually is quite common. We're seeing multiple females every single year are double clutching. Okay, June 2025, this is the next year. This is the final year of this study. We exposed 23 nests and had 6 failed nests, and again, all the site characteristics were recorded. So as you can see here, they were obviously preferentially choosing one side and also quite close to the water. That'll kind of come into what we're going to find out with the results. Here's another map. So this is the map of they introduced in 2025. They also introduced random sites in order to truly and accurately study nest site selection. So they recorded nest site characteristics at these sites to compare to the nests that the females chose. They also conducted soil sampling for all random nest sites in 2025 for soil moisture and bulk density. So again, we're trying to figure out, is nest site selection something they actually do, or is it a random event? Here is an extremely boring table, which I'm just going to skip over because I don't think this is fun for any of us to go through. So instead, I'm going to go right to the results. So we found for our results that the northwestern pond turtle nest non-randomly, with successful nests being placed closer to water and canopy cover in areas with higher soil moisture relative to random sites, and these factors that shape the incubation microclimate and survival of hatchlings. So contrary to previous assumptions, which again goes back to the first thing, previous assumptions, contrary to previous assumptions, nesting habitat, slope, aspect, grass height, and ground cover were not significant predictors of nest site selection in the study. Previous studies showed that nesting habitat have largely been descriptive and may reflect patterns of habitat availability rather than the active female preference due to a lack of comparing chosen site nesting characteristics with random sites. So, which I did not mention earlier and I should have, almost every bit of literature you'll read is that turtles prefer nesting on south-facing slopes. And in the study that we chose, the pond that we had, not one turtle nested on a south-facing slope. Again, showing that a lot of things are based on what people have studied in small subsamples and not what's actually happening. And so what we really wanted to show here is that um, the selection of habitat types is not based on an aspect. Instead, it's actually based on the female choosing correct aspects, such as uh, um, canopy cover and closest to water and soil moisture. So let's get back into this. So chosen nest sites tended to occur in soils with high bulk density rather than random sites. It's suggesting that compaction may further constrain nesting success under drier conditions. So therefore, if the conditions are really dry, it's going to be harder for the females to nest, and therefore the females will not select those really tough soils in really dry conditions. And that's probably why we also had so many failed nests, is that if a female runs into intense soil, intense compacted soil, she may try to dig through it for a few hours and then turn around and try somewhere else later. Oftentimes, she would not even return to the exact same spot, would actually choose a different spot with different soil moisture. And in general, bulk density decreases as moisture content increases, and therefore these factors likely influence nesting outcomes, which is why we saw so many failed nests. Sorry, give me a second. So in this context, the observed tendency for canopy selection to be driven by low temperatures likely reflects response that balances the need to avoid excessive heat while buffering against rapid nighttime cooling. What's important and cool is that the behavior, flexibility, and site selection may allow females to mitigate against thermal risks by shifting their nest sites to create a thermal buffer for their embryos. Similar patterns have been documented in other turtle species, which alter nest placement relative to distance from water and canopy cover in response to environmental conditions. So what we're saying here is that based on the study, turtles, instead of just selecting a single aspect, just a south-facing slope or north-facing slope, are actually choosing canopy selection. And the reason why is they actually control the temperature of their nests to avoid that excessive heat, which either would drive the population towards only females or actually end up in embryonic failure. And so it's very interesting when females are selecting that this turtle that's this ancient creature, which we'd all probably think is kind of dumb, are actually selecting very specific habitat traits in order to lay their eggs in order to best benefit their offspring. Here's a figure showing the trade-offs. So again, you see the majority of nests happening closer to water. Fairly basic little figure here. And again, this is the failed versus successful. So again, distance from water. So distance from water, this includes distance to creek as well. The closer they are, the more successful. Failed nests began to crop up a few more once they got further and further from water. So to summarize this before I move on to eDNA, nest site selection with respect to distance from water, distance from canopy cover, and soil moisture likely reflects a complex mosaic of trade-offs, including minimizing nest predation and cattle disturbance, maximizing offspring survival during dispersal back to aquatic habitats, ensuring survival of nesting females during overland movement, and responding to environmental variation that influences incubation conditions, sex ratios, and hatchling success. So therefore, females have the ability to, at least in the study, it appears, to select to mitigate for all the different things that can potentially happen to turtle, the overheating, the mass predation, all these different things. So turtles are selecting nests that are hopefully maximizing hatching success. These findings also have direct implications for conservation and management. First, the importance of proximity to both the stock pond and creeks indicates that connectivity among multiple aquatic features within upland landscapes is very critical. You can't just have a single pond. It's actually much more important to have creeks and ponds with drainages and all that stuff if you can. We have had instances of ponds we work on that are so isolated that you actually have the opposite effect. They actually can breed quite well. The turtles are kind of doing fine, but the issue is that it actually becomes a sink where it's actually overpopulated. There's not enough food for them. So having multiple different linkages of ponds or creeks to ponds is extremely important. In addition, creating fixed buffers around ponds, which is what we do for protection, that may fail to capture the full range of habitat used by nesting females. Nesting females don't just nest directly next to the pond. They actually can travel quite far, 100, 200, I think 150 meters was the farthest that Maddie saw. And so instead of setting a, say, 60-meter buffer, saying that's going to capture everything, and looking at the males, 60 meters will do nothing because males can travel, obviously, 6K is what we saw, 7K. Instead, they should actually prioritize protecting networks of aquatic features within upland nesting areas. So instead of just saying, we set this buffer around a pond, maybe look at the drainage, set a buffer around that, and think about things more than just, okay, 500 meters should cover most. And while it kind of does, covered a lot, it's not going to cover everything. So there's just not really a one size fits all. And every single time we create a new conservation practice or strategy to protect turtles, we should look at every pond or habitat type differently because every single situation is different than the last one. Second, the association between nest success and soil moisture underscores the importance of maintaining soil conditions that facilitate excavation and incubation. Minimizing soil compaction could potentially improve nesting conditions, such as reducing livestock pressure and nesting habitat during or after rain. So those are the main takeaways. Maintaining a nice buffer around a pond could potentially help, but I don't think it's going to cover everything. And really focusing on canopy cover. And if soil can be looser and less compact during the breeding season, that may potentially help. Although, funny enough, this was a heavily grazed area, well grazed, but it's heavily grazed. It's an active livestock pond. And we actually saw great reproductive success. So again, it's one of those things we think we know a lot and we're starting to learn more, but we still have a lot more to learn. I want to say thanks for this portion. We have a ton of collaborators. This is our Western Pond Turtle Collaborative, is our group in the middle. If you feel like getting a hat with a turtle cowboy on it, let us know. They're being sold right now. But thanks to everybody listed here, including Ben, who's on the line. Okay, so next part of this, eDNA. So over the past 4 years, again, Ben and I have been on this project of incorporating eDNA analysis into rangeland biodiversity research. I'm not sure how much you guys know about eDNA, so I'm going to give you the background first. So here's kind of a figure of traditional techniques. So we have a ton of techniques to look at what species are on the field, look at biodiversity. There's trapping, which is the most invasive. It's what we do with the turtles. It's capturing them, marking them, all those things. There's visual surveys, which is point counts, camera traps, drones even now. There's audio recordings. There's human ear. Merlin's the greatest app in the entire world. If you birdwatch, it's truly incredible. It's AI that tells you what birds you're listening to. There's traces like tracks and scat that does require you to know a little bit about things. But finally, there is environmental DNA. So environmental DNA is about a 20-ish, 25-ish year old technology now, but that's considered extremely new in the world of ecology. And what environmental DNA does is it allows you to detect DNA that is shed by an organism into its environment and collected environmental samples. So here I'm trying to show you everything sheds DNA. We're all shedding DNA as we sit here. If you touch your head, you're shedding DNA. If you scratch yourself, you're shedding DNA. So what environmental DNA is, it takes that off the ground, off water, whatever it is, and actually is able to process and tell you what shed that DNA. So again, DNA is everywhere. You can get it from everything. Even pollen will contain DNA, not only from the flower itself, but also from the bee that's pollinating it, or even the hummingbird. It can be anywhere, and you can detect it through anything. So, hair and fur, skin, cells, tissue, scat, urine, blood, pollen, everything. And so, what we do is an eDNA researcher, or what an eDNA does is we sample this through environmental vectors. So, poop, water, soil, vegetation, blood, which is probably my favorite 'cause it's kind of crazy, um, and uh, and air. Air is a new one, and they're starting to get some really good results where you actually can process what's in the air and detect what species are present. Um, so in collecting this, what do? You simply get your filter, whatever water filter, your air sample, your blood vial. You send it off to a lab, and the lab then extracts this DNA and in the laboratory, and then creates what is just a nice little vial of just DNA, and they analyze that sample. And so they do this in two different ways. qPCR, which is like your COVID test, where you're looking at a single thing. So if you want to know if a turtle's there, you can take a test. You can literally do snap tests now, where you can just say turtles are present, turtles are not. It's extremely sensitive, but again, it's very limited. It's just a single species or a couple species. Or there's metabarcoding, which is what Ben and I are doing. So this is a multi-species analysis. So this allows you to take a sample and look at groups. So say you're looking at fishes, or say you're looking at vertebrates or invertebrates. This sample can let you look at all those at once. And it's slightly less sensitive, but it's still extremely powerful and getting better every single day. So I'm going to focus on metabarcoding for this presentation. So again, the eDNA is processed, it's sequenced, and then it's compared to a DNA library to identify the organism. So from this sample goes through the whole process, and then they identify whatever it is. And then we can use that as a biodiversity indicator or just a presence absence. So I think the powerful thing about metabarcoding is you actually use it for biodiversity. So instead of just saying turtles are present or turtles are not, you actually will say all of these different species are present. So it gives you a really good idea, a strong idea of what actually is present and how that compares to other sites. So how do we use this data? Well, we use this two different ways. One is to detect the presence of species, including rare ones, elusive, dangerous, invasive, all the different things you can. And then you're also able to estimate the relative abundance. So if you have a lot of DNA, so say you have almost a ton of DNA from fish or one single species of fish and very, very small amount of DNA from CTS, you can get an idea there's probably a lot more fish than there are CTS there. It's not perfect. You can't say there's 5 CTS, 20 fish, 10 red-tailed blackbirds, but you are able to say there's a lot of DNA from this species, very little from this one, and therefore get an idea of kind of species abundance. Also, it's very interesting across a scale. So if you have, say, 20 ponds, 10 of those ponds have a very small amount, 3 of those ponds have a medium amount, and 2 of those ponds have a ton. All of a sudden, you can say, okay, these ponds have a higher abundance of this species than these other ones. But again, it's relative, so it's comparing amount of DNA, not individuals. And theoretically, it could just be a lot of DNA from one individual releasing at once, but usually it's a very good tell of relative abundance. Advantages of this versus traditional. The biggest thing is that you don't need to see it. So we as people may not see things, especially for kind of more elusive species like CTS. As you go out there, you bring your binoculars, your cameras, your nets, and everything, and you may never catch a CTS. I know a lot of people have gone out and have never actually seen one, and yet they are utilizing this waterway. So DNA allows you to see things that are not necessarily visible. So using DNA to find these kind of rare and elusive species. Um, they have been proven to, so there's been a lot of literature over the last decade looking at eDNA compared to other methods. It's kind of what Ben and I are doing with the NRCS right now, is trying to see how effective it is versus other things. So Advantage to Ben, this is a lot of scientific literature. So electrofishing, camera trapping, seine netting. All these studies show that eDNA was more effective. Rare and elusive ones, they actually were able to find the night parrot for the first time, which is a parrot in Australia using eDNA in little ponds. Goldie finches as well. And then invasive species are quite good at detecting early. It's another use of eDNA is as an early detector. So taking consistent DNA samples and using it as like the new mussel, the golden mussel. It's a great way to say, okay, there's no signs of golden mussel, and then if you could sample all the time, if you get some hits early on, it could be an early warning system. So before people see this tiny little mussel, this could be a nice early warning system for these invasive species. In addition, there also is the biodiversity detection, which we are using. And also, here we go. So here is a nice simple figure if you guys ever wanted to know how well it does against other methods. So you have eDNA on the far right side. You have detections of StipNets, visual surveys, and seine netting. And as you can see, the eDNA, including metabarcoding and qPCR, performed as well, if not better, detecting all our local herp species than did visual surveys and DipNet. They still did really great, but you can see the rare and elusive ones, the tiger salamander, they were not even getting detections with dip netting and visual surveys, and yet you could pick it up with eDNA. So it's kind of a nice paper showing just the power of this thing, especially for these rare and elusive ones. In addition, this is a study I took part in Australia. It's called the Great Australian Platypus Search. It's looking at eDNA on a scale, and so this idea of biodiversity. And so you could use eDNA with 350 sampling sites over 22 days, 1,000 eDNA samples. You're able to not just get, that's a lot of work. 350 days of sampling visually is a lot of work. Instead, we did this very quickly, and we're able to get 61 bird species, 36 mammal species, including platypus, 9 amphibians, 50 fish, 19 crayfish. So you're able to get these really robust species counts. Okay, limits. Go through this quickly because the eDNA is not perfect by any means. Um, there's a lot of limits. Sunlight breaks it down, changes in pH can affect it, microbes, enzymes, temperature affects it. And also you're dealing with like a 2 week window max. So if you take an eDNA sample, you cannot say the species is there at that second. It could have been there 4 days earlier, could have been there 5 days earlier. DNA lingers, so you're getting a bit more of a time window than you're getting this exact moment in time. It still is a moment in time in a sense because it's within that 2 weeks, but you are getting a bit of a range. Um, in addition, you get false positives. So if you're walking around, this is a funny one that we've had in our data, giraffes pop up randomly a lot. And the group we work with is able to link it back to a lot of times people going to the zoo or doing something else. So if you go to the zoo and you go back into a pond and you step in something that you shouldn't have at the zoo and go to a pond, you could leave DNA from an elephant or a giraffe at that pond. So you do get false positives of species that are not there, and you also get false negatives. So a species could be there, you just missed the DNA. This happens a lot with soil sampling because soil is fairly restricted. You got to make sure you really cover perfectly. But if you're trying to get a lion footprint and you know it walks this thing, but you don't select perfectly where the footprint is, you may not actually get that DNA. So ongoing work we have. So we have working with a Coastal Conservancy. We have CSP programs. Ben has a million programs as well. And we also have a SIG program. So we're at the NRCS. I'll skip over that because it's not fully up to date. We have a bunch of programs we're doing across Alameda and Contra Costa County, hopefully leading to something really cool because we have 3-ish plus years of data now. So one of these days, Ben and I have to write something up that would be really cool. So give you some live stuff we've done just to show you kind of the power of eDNA. So here is pond 26, the pond we restored a few years ago. So prior to us actually doing the survey, we went out there and did a couple different things with eDNA. So we went and did a traditional survey, we did metabarcode, we did qPCR. So qPCR, all we were looking at was red-legged frogs, and we saw red-legged frogs. So yes, if that's all we wanted to know, are the red-legged frogs present? There were. Fantastic. During that same time, we did a traditional survey. No one saw red-legged frogs. We did, however, see a cow, a red-winged blackbird. We saw tree frogs, and we saw a human. Go figure. But with metabarcoding, we took the same time, sampled the same filter, and we actually able to see all of these things. So it kind of shows you the power here of, while it's not perfect, this was able to, because of just the power of DNA, see a larger amount of species than just a simple visual traditional survey, which was a visual survey in HandNet, which is 3 people, by the way, in over an hour. And me taking a DNA sample, which was, I think it takes maybe 15 minutes, less really. Depends however quick you are. So it's quite power can be quite a powerful tool. Um, and again, just focusing on the two different ones, that shows kind of the difference in Medicart barcoding versus qPCR. Okay, so this is some other things. So this is the collection of methodologies to each regional parks. So again, just showing you using DNA, we're actually able to find across all of these ponds, we're able to get regulated frogs across all these and DNA. So this is kind of what a sample would look like. So we're able to see like the presence of all these species across. And not only that, but you also can get this was actually two qPCR tests we were doing. And not only were we able to get with qPCR 100% detection, which is green, you also get a middle and a light detection too. So if you have like a small amount of DNA or softer detection versus a heavy detection. So you can see a high amount being the green, where positive, this kind of softer green would get us kind of this like a good, it's still detected, but not a little bit less than the other ones. And then yellow it actually is considered technically a non-detection because it only detected one of three samples, but it does show that there is presence of DNA there, but not necessarily, the species may not necessarily be present. So part of this whole study and what we are looking at is we want to answer a few questions. So how does eDNA perform within livestock ponds? How does it compare to traditional survey techniques? And can we use eDNA to measure the success of a pond restoration? So here's what our DNA testing looks like. So we go on the field, we have a eDNA a citizen science collector. We collect about 5 liters in a bucket, and then we sample out buckets. We're actually able to take multiple points around the pond. So how does it do within livestock ponds? We have 2 years of data here. So across all these sites, which combines Ben and my sites, we were able to detect 82 species of vertebrates, 102 species of inverts. And if you break it down by the classes, you have 23 mammals, 8 amphibian, 26 birds, 23 fishes, and 2 reptiles, which is pretty incredible. So across the 2 years, you're able to get this huge number of species. I find always the most impressive is mammals. I can think of very few mammals I actually get to see in the wild on a regular basis. It's kind of us and cows and occasionally deer. Um, the fact that we were able to get 23, which then included, I know for a fact, bobcats, mountain lions, is kind of amazing.
The
Also, a
one that,
ton of,
yeah,
yeah, bats a 10. Sorry, God.
I was going to say the one that got me was bats. We found
Yep.
those out near Vasco that obviously we're not seeing those in the middle of the day. So knowing that they're using these ponds is kind of new information for me, at least.
Yeah. Oh, bats we see them everywhere, including troughs too. We take samples from troughs, and bats use everything. And I think it's kind of amazing. This animal that does not land in the water still sheds enough DNA from just flying over and sipping from it. It's pretty incredible. So yes, it can be used in livestock ponds and quite successfully. We're able to detect a ton of things. Here's a hot heat map, which I like to show just because it brags about how much better Alameda County is. Because look at all our hotspots compared to Contra Costa. But just kidding, just kidding. But it's kind of cool. You can use this data to really show where the highest species abundance is using these kind of basic maps, showing that Snow, and this is actually Pleasant Ridge, that's quite an abundance. And then down here is Ohlone, again, quite a diversity of species, including with, what would this be, Ben?
That's Diablo Foothills, I believe,
Okay, so yeah, Diablo Foothills, which I guess we've
and
had,
Shell Ridge. Yeah,
yeah, many years of sampling there too, so that makes sense.
yeah.
Um, and some highlights we were saying earlier: pumas, pond turtles, montane, so a bat species, dusky-footed wood rat, western tiger salamander. So a lot of these are actually listed species. Although you probably noticed at the bottom left, we did get sea otter. That was not there. That's one of those false positives we're talking about. There was an issue with some sea otter DNA in the lab, and therefore sea otter popped up in a lot of our samples. So again, it's great. It is able to show you some things that you would not be able to see, like a puma. However, you're going to have some issues, just like how there's observer bias and things we're going to miss more in the field because we're just humans. There also are issues with eDNA. Again, I'm going to show you this. This is just how it compares. So single species red-legged frogs versus metabarcoding versus traditional survey. It can really, in many cases, it can outperform traditional surveys. And also it really maximizes time. So 15 minutes versus an hour. And then finally, how do we measure success? So we were really interested in the idea of using this as a tool for success. Currently, no one wants to pay us to do monitoring. I'm sure you guys have all experienced in any restoration work you've done. It's really hard to get money to follow up and do monitoring checks. So what we wanted to figure out is for our pond restorations, which no one's really doing how well to do. It's more anecdotal. Like we do see red-legged frogs come back, we do see turtles come back, but are they What else comes back? How well are they doing? All that stuff. We wanted to see if DNA can be used as a sign of success. So if we sample before restoration, after restoration, and a couple of years after restoration, what does that look like? These restorations has been kind of we talked about a little bit. This was a pond that was absolutely inundated with cattails. We dug out the entire thing. We like scraped the thing up truly clean, and then we went back. This is about, I don't know. This is a year later almost. This is what the pond looked like. It's absolutely beautiful now, and we took eDNA samples. So pre-construction, we had California newts, we had Sierra tree frogs, California red-legged frogs, a duck species popped up, red-winged blackbirds, cows, humans. And post-construction, oh, this is 5 months later, we were able to already detect within just 5 months of cleaning this thing out. We mean, we're digging it down to its base layer. All water was drained out of it. We got California newts back, Sierra tree frogs, red-legged frogs pop back. We didn't have ducks or red-winged blackbirds. That's okay. Still had cows, and we also got a coyote this time. So within 5 months, you can see that these ponds are bouncing back. So even if you clean out a pond, um, you can really see these species coming back. And that's what the power of the cDNA stuff is. It gave us this ability to quickly go back, very little budget, get a sample, process it, and give us some evidence that our pond restorations are doing great. They do good things for the get-go, but also the animals do bounce back really quickly. Okay, that is the conclusion. So many partners as always. I can probably keep talking about more partners, but if you have questions, I included Ben's email here too. So if you want to talk about eDNA stuff, I put Ben and myself, and then also Julie Stanford, who's our eDNA Explorer person. So if you're interested in doing eDNA yourself, if you have a pond in mind, if something like that, either you can talk to us, or this is a citizen science thing. You can yourself can go out with a filter and a little syringe and take an eDNA sample, and you can get a process. So if you have an interested body of water, something you want to, a small little grant, if you will, you can, if you can get it, that's all these things that you guys can go out and do yourselves. It's very citizen science friendly, which is great.